All right. I want to welcome all of you to our special Antarctica Day celebration. We're really excited to be talking to Tim Dwyer and Art Woods. They're in McMurdo, Antarctica today, December 1st, 2016. We have a lot that they want to cover, including a little bit about Antarctica and why it's important for science and research and important to the globe. And we will also hear about the work that they've been doing the last 6 weeks, I believe, down there in McMurdo. So we're really excited to hear that. We're going to go through a few things before we turn the presentation over to Tim and Art. As you can see, we do have a webcam. The webcam is just going to be used for the presenters. And if you're just joining us, we're going to ask you to mute phones and mute your computer mics so that we don't get classroom noise. I know there's a lot of people joining from around the world today. What's happening now is that there is a slide in the center, and that's where the presentation is going to be. There's an area for chatting. And we will take questions there, and then we will, at the end, also have time for questions from everybody. As we go through the presentations, please introduce yourself, and then, like I said, post any chat questions down in the public chat area. This event will be archived, and we will post the archive online and share it with everybody that's registered. In celebration of Antarctica Day and the organizations that are hosting it are Spaces and Association of Early Career Polar Scientists and the Arctic Research Consortium of the United States. You can launch your virtual balloon and show that you are participating by going to this link. We'll put it up at the end of the presentation, so if you don't have a chance to do that now, that's okay. A few things about why Tim is in Antarctica to begin with. He's part of a National Science Foundation program called PolarTREC, Teachers and Researchers Exploring and Collaborating. And we place teachers from around the United States with researchers like Art Wood in either the Arctic or Antarctic so that they can get hands-on science experience and share that and bring that back to their classrooms throughout the United States. We've been doing this since 2004, and we're really excited to be hosting Tim with Art on this expedition as well. I already mentioned questions. We'll put the slide back up at the end, but as we go along in the presentation, please type them in, and we'll relay any questions that the presenters don't see as the chat goes as they present. So I think I covered everything. So with that, I'm going to turn this over to Tim and Art. And like I said, they are in McMurdo, Antarctica. The technology and the communications are sometimes sketchy, so we'll see if we can keep the webcam on and also if we can hear them. But don't be surprised if the connection gets dropped or they have to log back in. That's just the way of doing these things when people are joining from around the world. So welcome, Tim and Art. I'll let you take it from here.
Good morning. Happy Antarctica Day, and welcome to the Prairie Laboratory in McMurdo Station on Ross Island at 1178 degrees south. My name is Tim Dwyer. I'm a teacher at Twin Street International School, as Janet said, in Friday Harbor, Washington.
I'm Art Woods. I'm a professor of biology at the University of Montana.
Cool. Next slide. So, as you guys know, because everybody's here as a fan of Antarctica, Antarctica is the fifth largest continent. It's about 1.5 times the size of the United States. It's significant because 98% of it's covered by ice, which is several kilometers thick in places. It's got most of the world's fresh water locked up in ice. And it's also the highest continent because of those ice sheets. In a lot of places, most of it's over 3,000 meters above sea level. driest continent, actually a desert. The South Pole receives less than 10 centimeters of rain, of rain equivalent precipitation every year. And it's also surrounded by the Southern Ocean, whose currents and winds isolate it. Early explorers visited Antarctica starting in the 1820s. They were people who were coming down here to hunt seals. That was the first time the continent was sighted, and the first explorations of its coastline began in the 1830s and 1840s. The first explorations of the interior started in the 1890s. And this is a photograph of Robert Falcon Scott's Terra Nova hut at Cape Evans. This was the hut that the Scott expedition left from to reach the South Pole. Of course, the story everybody's familiar with is that they perished on the way back, but it's the pervasive operations that their expedition used. It is now a property, a facility maintained by the Antarctic Heritage Trust that's held in a high state of preservation as a monument to those explorations in the early part of the 20th century. The South Pole was reached by Scott in 1912. He was about a month late. Adamson and his team reached it in December of 1911. And way back in the late 19th century, when people were exploring the coastline, countries started to make territorial claims. And in order to make a territorial claim to Antarctica, really what they did is they explored and mapped the coastline and then said, we have everything to the South Pole. So that's why this map actually looks like kind of a pie chart. 7 different nations have made claims to the continent of Antarctica. But in order for those claims to be taken seriously, they actually have to be recognized by other nations. One of the things that you need to do, of course, is explore the region that you've claimed. But you also have to put a settlement there, which is a little bit tricky in Antarctica with average temperatures well below freezing much of the year, and actually no soil to speak of, or really any dry land in a lot of places. So nobody actually had settled Antarctica by the early 20th century. And a large part of those early explorations actually involved science. When Scott's team came back from the South Pole, and again were eventually starved to death out on the ice, trapped in a blizzard, the rescue party, or the recovery party that went back to find them, actually found that they were hauling geologic samples. That meant that they were bringing hundreds of pounds of rocks with them across the ice. And I bring that up because I want to illustrate the fact that science was very, very important to early exploration. So here is a photograph inside Scott's hut. Go ahead, Janet.
Tim? Yeah, it's hard to hear you, so you got to talk probably a little bit slower and directly into the microphone, and probably scream Art's ear off there.
All right. I'll talk a lot louder and a lot slower. Okay. How's that? Any
So
better?
far better. Thank you.
Okay.
Yes.
So this is a photograph inside of the Terra Nova hut. We were able to visit with our team here. One of our dive sites is nearby, and it's an illustration of the scientific equipment that was brought on these early explorations. This is from that 1911 exploration. In 1957 and 1958, during the Cold War, a group of countries got together to launch the International Geophysical Year, which was sort of a full-court press of science by 67 different countries that made coordinated efforts to understand the Earth. And they studied auroras, cosmic rays, gravity. They did precision mapping, meteorology, oceanography, seismology. All of these things were big efforts as part of the International Geophysical Year. Rocket science, the first satellites launched during the International Geophysical Year. And it was an astounding success. It promoted the exchange of findings and sharing of efforts during a period of, during the Cold War, which was not really known for international collaboration. There were some new discoveries, such as the Van Allen radiation belt, as well as mid-ocean ridges being defined, which was an important evidence for confirming the theory of plate tectonics. So, in 1959, 12 of the countries that had done work in the Antarctic during the International Geophysical Year decided to put together a treaty that would promote Antarctica in the interest, that would preserve Antarctica in the interest of all mankind, and that it shall continue forever to be used exclusively for peaceful purposes and shall not become the scene or object of international discord. So what that means is that all of those claims to Antarctic territory were neither annulled nor were they recognized. The Antarctic Treaty also prohibits the use of fortifications or armed forces here, although the military is used for transportation and science support. And there's no nuclear testing, no nuclear waste. Each of the countries has the right to inspect the facilities of every other member country. And there are annual meetings where diplomats of the signatory country closely coordinate efforts and share resources. From those 12 original countries in 1959, there are now 53 countries. And Antarctica, each of those countries has laws that support the Antarctic Treaty. So in the United States, an example of this is our Antarctic Conservation Act of 1978. Those laws apply to United States citizens. Over the years, there have been addenda to the treaty. One of them is the Convention for the Conservation of Antarctic Marine Living Resources that recently designated the Ross Sea area as a marine protected area. The Ross Sea, that comes into force next year. And as well as the Protocol on Environmental Protection to the Antarctic Treaty, which did a number of different things for protecting the environment in Antarctica. It allowed for or mandated environmental impact assessments. So environmental impact needed to be studied before anything went forward, any development plans went forward. Waste was managed. Marine pollution prevention was a big part of it. Non-native plants and animals were prohibited. So I've got a photograph of PolarTREC teacher David Deakinga here when we were in Christchurch, New Zealand on polar days there at the USAP Center. And he's posing with Kaya, a husky, who is far too young to be one of the last sled dog teams that left Antarctica in 1994. So we can't even have dogs here, no non-native species. And it also established protected areas. Specially protected areas, Antarctic specially protected areas, are one of the ways that research is influenced by the Antarctic Treaty. They are areas of special scientific or historical importance, and you need to have a permit to enter a special protected area. So here is our team visiting the Adelie penguin colony at Cape Roy, and we can't really get close to the colony at all because we don't have the correct permits for that. We're studying sea spiders, these are birds. We don't have a scientific interest in these animals other than that they're really cool and cute and adorable. So we've got to stop at the line there, and that's one of those signs that we see on our way into the atlas. So every year when diplomats get together at these consultative meetings, the Antarctic Treaty, there are a few organizations that provide an advisory role and help coordinate and progress the science. These are the Scientific Council on Antarctic Research, or SCAR, and the Council of Managers of National Antarctic Programs. So a member of the Council of Managers and the manager of the United States' National Antarctic Program, the USAP, is the National Science Foundation, which is the government agency whose mission is to advance the progress of science by funding scientific research and education. So NSF runs USAP, and that's an example of a National Antarctic Program and managers of that. Next slide. Okay, so here's a map of all of the research stations in Antarctica, both the year-round ones and the summer-only ones. And circled down there, kind of at the bottom of the map, is McMurdo Station, where we are right now. We're in the Ross Sea, right at the edge of the Ross Ice Shelf, which is the floating glacier that prohibits water access farther south. Next slide, please. This is McMurdo Station. It's the logistics base for the U.S. Antarctic Program. There are about 1,000 summertime residents here, and it's kind of like almost like a spaceport from the future. There's planes landing all the time, people going out to field camps, people coming in from field camps. To the other station, there are C-130s that go up to South Pole. It's a busy place, and it really is a hub. So that's where we're based, in the McCrary Laboratory and out on the sea ice around here. And I'll turn it over to Art to tell you a lot about our project.
Thanks, Tim. I'll try to keep talking loud too, but let me know if I fade in and out. So, just for those of you who joined late, I'm Art Woods. I'm a professor of biology at the University of Montana. And I've had a long-term interest in marine biology and in polar biology. And I first came to McMurdo in 2006 with some of the folks on my team today who I want to introduce now. So I'm working with this guy on the left is Brett Kowalski. He works on biomechanics, primarily of bird flight at the University of Montana, but he's down working on the biomechanics of sea spiders with us. The guy on the right is Steve Lane. He's a graduate student in my lab who's finishing up his dissertation in the next year, so quite a nice way along. I'm also working with Amy Moran, who's here looking through the microscope. She's a professor of biology at the University of Hawaii, so quite a temperature shock for her. And that's her student, Caitlin Shishido, in the background, and she's here working on her dissertation research also. So we're here to study an incredibly cool pattern called
Art?
Gigantism.
Excuse me, Art?
Yeah.
Yeah, so similar to Tim, they're having trouble hearing you. So if you talk slower and more directly into the mic, they'll be able to hear you better.
Okay, I'll do it. We are here to study PolarTREC, which is a pattern that's been known for about 100 years, ever since the explorers came to Antarctica. And it's a pattern in which animals that live in the poles tend to be much larger bodied than their relatives that live elsewhere in the world. And here's an example. So the insect on the right is an isopod, like a roly-poly that you might see in North America. The one on the right is a giant Antarctic isopod, and I have one in the tank right next to me. Just so you can get a view. So this is what I'm showing on that slide. It's called Glyptonotus, and it's one of the really big polar giants that lives in the ocean, just right offshore here. And we see it almost every dive we go on under the ice. Polar gigantism is important because we see it in a bunch of different groups of animals. And what this slide shows is a list of animal phyla. You may or may not know what an animal phylum is, but there are really major different kinds of animals in the world. There's about 35 animal phyla altogether, and this list here shows the ones in which we find polar gigantism. There's even some that are not even animals. So that that thing in the lower right is a single-celled organism called a foraminifera, and they're single cells that are so big that you can hold them in your hand and look at them. Cells this big around, so really amazing. We're here studying in Antarctica because the Antarctic continent itself is like a natural laboratory for evolution. It's been isolated for tens of millions of years from the rest of the world. It's been stable and cold. The water around Antarctica is about minus 1.8 degrees Celsius year-round. That's about 28 degrees Fahrenheit. And the things that live in that water have evolved special physiologies and special behaviors that allow them to live in such extreme cold. So it's like a sort of natural extreme laboratory, and we get a chance to come here and study the amazing things that live in it. The other reason to study gigantism is that there's some worry by biologists that the biggest bodied species may be the most vulnerable to climate change. So it's clear now that the world's oceans are warming up, and there's a lot of concern among biologists about what's going to happen to different animals that live in the ocean. And there's just a number of papers that have come out recently that suggest that really large-bodied organisms may be some of the first to be impacted by climate change. So it's important to, in the very beginning stages of this climate warming, to study the physiology and their evolution and their behavior. So we're working on a pretty simple set of questions that I'm listing here. The first one is maybe our main question: why are Antarctic giants so big, and how can they be so big? Why are they big here and not big elsewhere in the world? We want to know how well sea spiders can tolerate changes in temperature and oxygen, and how temperature and oxygen interact with one another to affect the physiology and the evolution of body size. And we'd like to know whether these giants are going to be the losers in a warming ocean, more so than anything else. So we're studying a sort of poster child of polar gigantism. They're giant Antarctic sea spiders, and I'm holding one here. And this is really astonishing because most places in the world, I'm holding my fingers apart, a sea spider would be about that big. And down here, we see these really giant sea spiders all the time. And this one's almost the size of a dinner plate. And it's just unbelievably large for an organism like this. We are studying these sea spiders because they're one of the most dramatic examples of polar gigantism. And they're really common around here. They're easy to collect while we're scuba diving, and to bring back to the lab here and to do our physiological and biomechanical work on them. Just a quick overview of sea spider biology. So they're, as you saw in the one that I was holding, they have an extremely reduced body and really long legs. A lot of them, they have about 8 legs. And what's really weird about them is that all of the organs in their bodies are distributed down into their legs. So here's a picture that Tim took a couple of weeks ago of one of the common species around here. You see the legs up in the air, and if you look really close at that femur, that's a segment of a leg called a femur, you can see that it's packed full of eggs. So the female sea spider, she's got eggs in her legs, and she's got eggs in all 8 of her legs. The males have testes in their legs, and they also have extensions of their guts that go down to the very end of every walking leg. And I'll talk a little bit more about that in a second because we discovered something really interesting about the way their guts work. The spiders themselves are, the technical name is Picthingonid. So they're in the class Picthingonida. Depending on who you ask, there's some controversy about this. There's over 1,000, so maybe up to 2,000 species in the world, and a pretty significant fraction of those are Antarctic. Almost 200 species are known from polar waters, which implies that this is an important place for them, ecologically speaking, and that there's been evolution of sea spiders around the Antarctic continent. They are related to spiders in the sense that they're all in the group called chelicerates. So spiders are chelicerates, sea spiders are chelicerates, but they're incredibly distantly related to one another. The sea spiders are ancient, and their lineage broke off from the rest of arthropods about 500 million years ago or so. And so we like to say a sea spider is related to a normal spider that you would know, like a seahorse is related to a horse. That's about the sort of distance of that relationship. Their physiology that's relevant for us is that they have no respiratory organs, so no gills or lungs or anything like that. They don't breathe water in and out of their body. The oxygen just goes straight across the cuticle, and their blood is very simple. It doesn't have things like hemoglobin, like our blood does, to carry oxygen around. There are a number of projects that we're working on. I'm not going to talk about all of them, but we're a diverse group of biologists with diverse interests, and each of the members of the team, there's 6 of us all together, including Tim, have different sets of interests and expertise, and so we're working collaboratively on a set of separate projects, but that sort of overlap and give us an overall picture of spider biology. I just want to show you one result that we recently got, and this has to do with the question of how do you build a really big sea spider, and what they have to do differently if you're building a big sea spider compared to a small sea spider. And we discovered something really interesting about the way the cuticle is constructed. So the cuticle is the hard part. The stuff that I'm holding here, the red is the cuticle, and it's everywhere in the body. That cuticle provides the structural support so that they can walk around. And it's also true that the oxygen gets across the cuticle directly from the seawater. And to do that, it has to go through little pores in the cuticle that sea spiders can evolve. This picture on the left shows what a really small-bodied sea spider's cuticle looks like. It has just a few little dots, and those dots are pores. If you look at a medium-sized one, they have a lot more pores. And if you look at a giant one, they're basically like Swiss cheese cuticles. And we think that's because when they get really big and their cuticles are really thick, the only way they can get enough gas across into their bodies is by having these incredibly porous cuticles. So that's one way you can build a giant sea spider. Just a couple of minutes of info about the ecology of sea spiders, and then we'll wrap up our part of the presentation. We see them all over Eduardo Town wherever we scuba dive. This is a really large one that is shown in the foreground. In the background, you can see the sea ice that's frozen on top of the ocean, and that light thing is a crack in the sea ice that the light's coming through. We discovered that they're mostly scavengers in the ocean around here. So the big ones walk around and they look for other soft body organisms, things like jellyfish or anemones that have been trapped in some way or are damaged. And so here's a picture of a sea spider pulling a little chunk of flesh off of an anemone, off of a jellyfish that's been trapped on the bottom of the ocean. And here's a group of 4 of them that have coalesced around a jellyfish, and they're sort of slowly, very slowly, because it's cold, pulling it apart. So we think, actually, this is gives us a little bit of insight into why it might be good to be large as a sea spider. And I think the answer is that the large ones can move a lot faster around on the floor because they have a longer leg, and they can just simply go way faster than a small sea spider like this. And that helps them get to resources on the seafloor that they might want to eat before the little guys get in there and start eating it. Last quick thing about something we've discovered in just the last month that I'm really excited about. One of our questions has been, how do we get oxygen distributed in the body of a sea spider, right? They have to have oxygen in their mitochondria. And we'd like to know the mechanisms by which that oxygen moves around inside the body. And I personally have been working on a set of experiments that have tried to distinguish whether or not diffusion is enough. So maybe their long, skinny legs and the short distances is good enough just so that diffusion alone can let oxygen get in. They do have hearts. They have very small hearts in the middle, but they're very weak and they beat very slowly. And it turns out, we discovered, I think amazingly in the last month, that the main thing that moves liquids around inside their body is gut peristalsis. So this small sea spider you can see is transparent enough that you can see tubes in its legs. Those are the guts. And those guts have a very distinct way of peristalsing and moving the fluids back and forth in the body. And what we've discovered is that that is their respiratory organ that moves oxygen around. So in effect, this organ that originally evolved to do just digestion has been co-opted basically by evolution to help pump fluids around in the body and move oxygen from the tips of the legs where there's a lot of oxygen to the middle of the body where there's not very much oxygen. So we're still in the process of analyzing some of the data from that experiment. The experiments we do here, we're in an aquarium room now, and we have a lot of sea spiders here in seawater tables, but there's a really excellent set of labs here at the building. Here's a picture of Tim measuring the metabolic rate of the sea spiders in some little chambers that we made. He's standing in a room that's designed to be about minus 1.8 degrees C, so about the temperature of frozen seawater. That's why he's wearing his big red jacket. And just here at the end, we do do scuba diving to collect our spiders. There's a team here that drills holes through the ice, and we wear dry suits, and we dive under the ice. Here's a picture of Amy doing an experiment on the bottom under the ice where she's measuring how fast sea spiders move depending on their body size. And we do things like put out sensors to measure the ambient conditions, so how fast are the currents moving, how much oxygen is there in the water, what temperature is it. We're pretty sure we know what temperature it is, but it's possible there could be very slight variations, and so we measure that also. So we've got a ton of support from NSF and from the Curry Lab and the USAT, the U.S. Antarctic Program. And it's just amazing to be down here doing science in such a remote location. As Tim said, it feels like we're on a different planet. And yet, here we are in this well-equipped lab and able to do really top-notch science. So it's been a great experience. That's all we have. And at this point, we'd like to turn it over for questions for either one of us.
Yeah, so thank you very much, you guys. We have a lot of questions. Can you hear me okay, Tim and Art?
Yep, hear you loud and clear.
Okay, great, because we got a whole slew of questions. So I'm going to back up. One of the first questions that was asked was about have you witnessed any climate change in relationship to the sea spiders and populations down there underneath the ice? Do you see climate change?
So Antarctica is relatively slow to change compared to the Arctic. So if you look at temperatures in the Arctic, they're rising sharply. They're not rising sharply here yet, and right now the temperature of the seawater is still right at the freezing point of seawater, so 28 degrees Fahrenheit. But there is another effect that climate change is starting to have, and that is the increasing levels of carbon dioxide in the atmosphere. That carbon dioxide dissolves into the ocean and lowers the pH of the ocean, makes it more acid. And there's been a group here for the last few years studying the effects of that increased acidity on the shells of organisms that make hard shells. And it turns out that the acid appears to be eating away at the shells a little bit. And so there's some worry that the early effect is going to be from changes in ocean pH rather than changes in temperature. But over the longer term, we're also going to have increases in temperature.
Okay, great. So I'm going to just start with all these other questions. A lot of them came from students. Let's see. We have, what is the biggest sea spider you have seen?
We got one that was 22 grams. A little hard to envision what that means, but here, you show it to him. Its leg span was about like that. And it was so charismatic that we named it Pinky. So this is one of the only sea spiders that got a name. And we delivered Pinky back to the place where we collected Pinky about 5 days ago. So Pinky is back crawling around the bottom of McMurdo Sound. And we initially thought Pinky was a boy, and that's why we called Pinky Pinky. Turned out Pinky was a girl. And so we changed the name to Pinkalicious.
Nice. This one is for Tim also. Do you feel cold when you are scuba diving?
Yeah, actually, we do feel cold when we're scuba diving. We wear dry suits and gloves and hoods and layers and layers of warm things underneath. But even after about half an hour or 45 minutes, that cold gets to us and our hands become difficult to use. It's time to get out of the water around then.
Yikes. Okay. From my class, would gigantism appear on land if oxygen levels were higher?
Oh, that's a great question. And there's been some really interesting work done on that. About 300 million years ago, the oxygen levels on Earth were higher in the atmosphere, and the geochemists think that they might have gone up to 30% or even 35%, and as opposed to what we have today, which is just 21%. So a lot more oxygen 300 million years ago. And at that same time, we saw the evolution of really giant insects on land. There were dragonflies that had wingspans like this, and that weighed, many, many grams. It's called Paleogigantism, and there's a number of people in the world studying this relationship and trying to figure out if more oxygen over history led to the evolution of big body size. So, and the jury is still out. We don't know for sure, but it's a tantalizing pattern. So good question.
Okay, several questions about the reproduction and why they carry the eggs in their legs. People miss that. So, and also how many eggs. So, can you kind of refresh us on that piece?
Yeah. So there are two sexes. There are boys and girls, which is not true of all marine invertebrates. Some of them are asexual, some of them are hermaphrodites, which means that they have simultaneously ovaries and testes in the same individual. In sea spiders, because their abdomens are so reduced, there's no room for anything inside them. And that basically has forced all of the organs out into the legs. And so because they have no space anywhere else in their body, they make sperm and eggs in their legs. And when they mate, they come together and they line up their legs, and they have little holes on their legs, and they get the sperm and the eggs together. Another really cool thing about this is that the females, once the eggs are fertilized, she gives the eggs to the male, and the male carries them around while they develop. It's a little bit like the way seahorses do it. And what I'm showing here is a male sea spider. If you can see these little orange, this orange stuff right here, that's a couple of egg masses, and it'll hold on to those eggs for maybe, we don't even know how long, but probably 8 months or a year or even longer while the babies are developing. And the males have special arms that they use just to carry those eggs around. So yeah, really interesting. One
Cool.
of those genus, one of the most common genera here is called Colossentai, and it doesn't have egg masses. And one of the big mysteries is, how does it have babies? And we've been trying to figure that out while we've been down here, and we haven't done it. But one idea is that the babies, once the eggs are fertilized, they just float away into the plankton, and the babies grow up swimming around in the plankton like a lot of the other planktonic organisms do.
Cool. Okay. So yeah, when you show us the spiders up close, we can actually hear you better. So I think you just need to show us the spiders up close all the time.
Okay, good.
Okay. So Erin Lynch, whose second grade class, says they are loving this. They just wanted to relay that to you. Another question from Jeff's group is, do you know how many sea spiders there are in Antarctica?
How many sea spiders there are in Antarctica? Well, there are about 190 different species of sea spiders in Antarctica. I know that. And maybe about 250 in Antarctic and sub-Antarctic. But as to their total population, no, we don't know that. There are many things that we don't know about sea spiders. One of them is how many there are. We know that they go from shallow water to very deep water. We know that we find them all around Antarctica, but in terms of like their overall population, no.
Very large, like hundreds of millions or billions. And I guess if you're a invertebrate biologist like me, that makes you really happy. If you're sort of on the fence about spiders, that may not make you very happy, but really amazing. Another question we get is, how old are they? And again, we don't know. We say that a lot to people that when they ask us this question. And they develop incredibly slowly. They grow up to adulthood very slowly. And it wouldn't be surprising if they live for 10 to 50 years. And some of them, the really big ones, we think maybe 100 years old or more. So really cold, polar, old giants.
I guess kind of related to the population question is from Sue Steiner, how many sea spiders do you see on average?
I would say on a typical dive, we would see maybe 100 sea spiders among the team. So we usually have 4 people in the water at the same time. So 2 of them are buddies with each other, the other 2 are buddies with each other, and we swim around and do our casts. And usually there's maybe 50 or 100 sea spiders. But there are a couple of spots that are sort of hot spots for them. And one of them is just right offshore here. And there we can see thousands of sea spiders. There's some kind of small ones that live in the mud that are just, they carpet the floor of Roberta's Mountain. Yeah.
Okay, we got several questions related to diving. One was, how long do you stay under the ice? And somebody just said, what are some of the precautions you need to take to stay safe diving?
Great question. So we stay under the ice as long as we can stand it, but it's usually not very long. About 45 minutes or so is maybe a couple of our team members did an hour-long dive yesterday or 2 days ago. I thought they were crazy. Also, you know, how long our air lasts as well, and what our nitrogen loading time is. And some of the precautions that we take to stay safe while diving, there's only one way in and one way out, and that's something to really consider. So we actually will hang a line with a weight on it that has a little strobe and checkered flags on it. So that's sort of like an upside-down radio antenna underwater that's sort of flashing light in the distance. The water here is very, very clear. We can see hundreds of meters, and we can see those strobes pretty far away. Not that you really want to get that far away from the hole. We always dive in pairs, So we're talking about a dive buddy. That's actually like a real technical term, diving partner. And we carry double regulators for each of us. The regulator in the super cold water, the thing that takes the air out of the tank and brings it down to a breathable pressure, can sometimes freeze up, and that actually just lets air out constantly. So we have a backup regulator on each of our tanks, and we always dive in pairs. We have oxygen kits available nearby, and on the station here, we have a recompression chamber. So if we were to have a dive accident, wherein we would have we got the bends or a similar or a pressure-related malady, we have a hyperbaric chamber here to treat those injuries. It only happened, I think, 7 times in the history of the U.S. Antarctic program, and hopefully not so. But it's good to know we have that safety net.
Yeah, that's great. I have some questions also for you, Tim, that are different. They aren't about diving or sea spiders. They're more about your excitement and being down there. So from Megan C., how do you think this experience will help your students, and what are you most excited to share?
You know, to me, I teach environmental science, and I think I look at McMurdo Station as kind of a microcosm of our planet in a lot of ways. Everything that's here had to be brought here, and because of the rules about environmental protection here, everything that's here has to be taken away eventually. So we can't, it's neat to think about what our planet would be like, or how we would have to change our lives around if our planet couldn't absorb any of the waste that we put out. So all of our waste food, all of our scraps, for example, get put into shipping containers and frozen and shipped back to the United States where they're incinerated as a way of keeping invasive species out of Antarctica, which is crazy and really expensive. But you know, so what does our planet provide us in that ecosystem resource and that ability to break down garbage? We can actually measure that, and I'm looking forward to bringing that to my environmental science class and starting to do some back of the envelope calculations on what these ecosystem services provide us that we take for granted in the rest of the world, but here in Antarctica is very much a thing that needs to be planned for and managed. Great question.
Okay, yeah, good. So there was another question that says, from Josh, Tim, would you briefly describe how you felt when you saw your first giant sea spider?
Yeah, it was, I was sort of actually aghast at how large they were. We didn't, some other divers had gone out and brought them back to us initially, and I just sat there watching them and watching them move. Again, in Friday Harbor where I live, they are like this big. And in fact, when Art and the guys from Montana came out this summer and did some diving up there, they actually had to pull them out. They were tiny. So I was really just sort of blown away, and they're sort of mesmerizing. You can just sit there and watch them just sort of like go about their work. They're very like, I don't know, they seem sort of like methodical elephant-like in a way, just sort of hanging out, moving very slowly. Yeah, they're neat to watch.
Okay, related to that, these questions are for Art. Art, when did you first become interested in studying sea spiders? And I think also related to that, you can, I'll ask you a question about science techniques. Are they similar to the techniques that early explorers or scientists use for your research? So start with why you're interested and go into techniques.
I came down to McMurdo for the first time in 2006 to work on polar gigantism in another group called nudibranchs, which are snails that live in the ocean. And we worked on nudibranchs, and I like nudibranchs a lot. But it was basically instant love when I saw the giant sea spiders. And you know, like Tim said, you swim around underwater, and they're just totally obvious. I mean, they're all over the bottom, and they're very clearly an important part of the ecology, and you know, it's sort of nerdy love for a biologist. You get into a group and you really get into it. And so at that point in the mid-2000s, we started thinking about trying to come back down to McMurdo to work on giant sea spiders, but we had other stuff to do in the States. I also work on insects back in the States, and we were working on some other projects. So now we finally came back last year and this year to work on this group that is just so incredibly charismatic. So, you know, I'd say seeing is love, and you know, it's important to just see these things in nature doing their thing. And for almost any group, you can find really amazing examples like that. In terms of the techniques we use, you know, science has come a long way in 100 years. You know, it's a funny mix of things here. So we bring down fancy equipment, much fancier equipment than anybody had previously. For example, I have a machine that can measure oxygen in water using little spots that sense the oxygen and a little laser that looks through the glass container that spot is in. And you really couldn't even do that 6 or 8 years ago very reliably. So we're sort of riding the technological wave as scientists make better stuff. But I would say at the same time, you know, we're also doing a kind of discovery science, which is actually what the explorers were doing 100 years ago, in the sense that there's been so few people here working on the biology that almost everything you do is completely new to science, and it's easy to find out fundamental new things. In one example from our work in 2006, we discovered 2 new species of nudibranchs, 2 new species of snails. And we weren't even trying. We were just scuba diving and picking up snails off the bottom. And a couple of them we brought back and said, we have no idea what this is. And the taxonomists eventually got to look at it and said, nobody's ever seen that species before. And they wrote a paper that described that new species. So most places in the world you can't do that because people have been around for a long time. But here it's just kind of wide open. I think this kind of discovery science is going to happen for a long time at the pole.
Great, thank you very much. Okay, so we're going to go back to sea spider questions, which there are no shortage of right now. And we have, let's see, you like them? So why do they molt, I guess, is one of the questions. Do they molt?
They do molt. So they're in a group of organisms, the arthropods, and even some other relatives that are outside of arthropods. And their defining characteristic is that they shed their exoskeleton every so often as they grow, and they make a new one underneath. So they do molt. One big question for us is whether they keep molting when they're giants, or when they grow up to giant size, do they have some final molt, and that's it. They're done for their entire lifetime. And we still don't know, and I guess we'll have to come back and figure it out. The really giant ones that we've seen often have many things growing right on their surface. So they might have bryozoans or barnacles sticking off of them, or diatoms and algae growing on their, Tim's going to go get an example, growing on their cuticles. And so we think one problem they face if they molt really infrequently or not at all is that they get too much other stuff growing on them, like an entire community of organisms on them, and that they have a hard time then breathing through their cuticles. So this picture shows a sea spider, this white stuff on it, those are barnacles, and so this thing is just completely covered in barnacles, and it looks to me like it would be really itchy. I have no idea if sea spiders feel itchy, but they're parasites, and they've hitched a ride on this sea spider, and we think they cause a lot of problems for the sea spider. And really the only way the sea spider could get rid of these is by molting and getting rid of its exoskeleton and starting fresh. The question is, can it do it? And we don't know.
Okay, cool. Thanks again. I was trying to show everybody your picture there when you got up close. let's see. Some more questions. Are sea spiders or other gigantism species endangered?
Well, they're not endangered in the sense that you might think of like a mammal in North America being endangered. You know, things that are endangered often have a total number of individuals in their species, like less than, say, a few thousand individuals or less than a few hundred individuals. There are no sea spider species that we know of that have that few individuals. But we're sort of approaching that question in a different way and asking, based on what we know about how they operate physiologically and how they respond to temperature and how they take up oxygen, can we make any systematic predictions about whether the big ones or the small ones are more at risk from climate change? And I would say our results are pointing toward this idea that the big ones are particularly vulnerable. You know, are they endangered now? No. But if you project out into the future and think about what's going to happen to the ocean, then there's certainly cause for concern.
Okay. So let's see. We got a question here about how do sea spiders communicate with each other, and also do they bite?
Yes. So we really don't know anything about the way they communicate, and they must communicate, right? Because the males and the females get together and mate. They don't make any sounds that we know of, although we haven't taken really sensitive microphones up and listened to them up in the water. My guess is that they communicate. They have lots of little sensors all over their legs, and they probably use those to smell or taste each other. And they also have little eyes on top of their head. They have a little, like a little thing that pokes up on the top of their head, and they have eyes that look in all directions at the same time. And so they probably are looking for each other also. They don't bite. They've never bitten us, and that's probably because they have a really tiny mouth. At the very end of what's called the proboscis, which is a big fleshy thing that sticks off their head, they have three little jars that are sort of triangular shaped, and they would walk up to let's say Tim is an anemone. They would walk up to the anemone and put their mouth on the anemone and start to grind with it. And that's about all that Tim would feel, is a little bit of grinding, and then they would pull a piece of Tim away and take it away. So we also are asked sometimes whether they're venomous, because they're chelicerates and they're related to actual spiders. We don't know that either, but I doubt it.
All right. We have a few more questions, and then we'll have to kind of sign off here. We've already been on for an hour, but let's see. From Andre Willie's class, can sea spiders survive on land?
No. There are no known terrestrial spiders, but that's a good question. There are many groups of invertebrates that have colonized, at least one species has colonized land. You know, for example, there are land crabs that spend all of their time on land and out of the sea. No one's discovered a terrestrial sea spider yet, and I guess I'm skeptical that they exist, but who am I to say?
Okay, and here's a question for Tim from Robert Dwyer. Why are some of your fingernails red? Is this damage from cold or repeated pressure exposure?
Thanks, Dad, for that question. Yeah, so that was morale. Actually, it was Thanksgiving here at Negredo Station. It's sort of a big deal. The people who work here, because it's so expensive to bring everybody down for all the way down here to work, they tend to work longer shifts, and they work a 6-day work week. So it's a 9 or 10-hour day, 6 days a week. And they had sort of an arts and crafts fair following Thanksgiving, and someone who's come out with us as a dive center will often bring out, or back up again, we'll often bring people out to our huts, to or don't get off stations very often. They're in a dining hall or in a cargo facility. But anyway, this person was trying to paint fingernails as part of the art and craft there, so I said, sign me up. And I just haven't gotten around to finding some acetone to clear it off. So no, I'm not injured in any way. Thanks for your concern, Dad. They've been taking pretty good care of me.
That's good to hear. So I think that's all the questions we're going to do for right now. I just wanted to find out from the team, what's your schedule? When are you going home, and what are you doing in the next couple of days before you go home?
Sure. When are we going home is to be determined. We thought we were leaving tomorrow. I think it turns out there's actually no flight scheduled for tomorrow. So it's Friday here in New Zealand time and Antarctica time. We have to pack up all of the experimental equipment. We've got to shut down the lab. We've got to clean our dormitory rooms. We've got to pack up all the cargo and ship that back. We've got to go through the actual sort of sign-out process. What else do we have to do? About it. Yeah, I think just everything. So our next couple of days are going to be sort of a hive of activity of packing up and shipping out.
Yes, I imagine you're pretty busy. Everybody is saying thank you, and they say safe travels home. It was a great presentation. Thanks for sharing your research with you. From Kim and Savvy, thank you. We enjoyed our time with Art and Tim. Safe travels home to both of you. While people are saying goodbye, I want to remind you to launch your virtual balloon, and also before you sign off, please fill out this poll. We want to thank our spaces and the APEX program and ARCIS for hosting Antarctica Day. This webinar is one of many activities that's going on, so make sure you visit the website to see what else is going on. And it was a really good introduction to Antarctica and to your science, and really fun to see live spiders all the way from McMurdo. So thank you very much. Great job, Art and Tim. It's been really cool. And I think that's it on my end. Anything you two want to say in closing?
Thanks everyone for listening in. It's really nice to have people interested in our work, and appreciate you listening to what we have to say. So thanks.
All right, great. With that, I also want to thank the National Science Foundation for funding PolarTREC, so teachers like Tim can go down and work with researchers like Art. It's fantastic to learn about places in the world that many of us won't ever get to see, and we're really glad. Tim, you've done an outstanding job of communicating the science throughout this whole process, and I hope you guys have a safe trip home. And with that, I'm going to stop the recording, and people or family that want to say hi, you're welcome to do so after I take the recording off.